each muscle fiber is supplied by a single axon terminal. Okay? And that's going to control that axon terminal, nerve ending, is going to provide innervation to that fiber that brings about contraction. Contracting muscles require continuous delivery of oxygen and nutrients. That's the purpose of the artery to that muscle. And then the waste have to be removed by veins. All right. Now, skeletal muscles span at least one joint. Some span two joints. And so they're typically attached to bone on either side of that joint. Okay? So, for example, let's talk about the brachialis muscle. Okay? This may not be exactly how it looks, but this is the humerus. And I've drawn kind of the ulnar right here. And the brachialis is going to span this joint here, which is the elbow joint. Okay? So the brachialis is going to extend from the humerus here. Oh, look here, it's going to attach to the coronoid process. Okay? So that when it contracts, it's going to cause flexion of the ulnar at the elbow joint. So that the angle of the elbow joint narrows. Okay? Now, the attachment to the bone that moves, in this case the ulnar, is called the insertion. The attachment, on the other hand, to the bone that does not move, in this case the humerus, is referred to as the muscle's origin. Okay? So the origin of the brachialis is the humerus. Its insertion is the coronoid process of the ulnar. Now, this is an example, by the way, of an indirect attachment. What do I mean indirect? Well, this muscle actually ends like this, and its tendon is what attaches to the bone on both sides. Okay? So it attaches via a tendon, a bundle of dense regular connective tissue, at both sides, at its origin and at its insertion. So that's referred to as an indirect attachment. There are some muscles that attach directly to bone without the need for a tendon. That would be referred to as a direct attachment. Look over here, for instance. Let me see. Oh, I see it over there. If you look at Red Fred's chest, his chest wall, okay, you see these intercostal muscles that span the space between an upper rib and a lower rib. They're attaching directly to bone without a tendon. That's an example of direct attachment.
Are there any big muscles that attach directly, or all your?
You know, I can't think of any immediately. Some of the big muscles use an aponeurosis, but that's still an indirect attachment.
So the intercostal.
Oh, here's that brachialis muscle we're talking about. Okay? Such that when it contracts, there's movement at its insertion, the ulnar. And with movement of the ulnar, the elbow flexes. There's flexion of the elbow. Now some muscles rather, span two different joints. In fact, that's the case for the biceps brachii, the famous biceps muscle. Okay? Part of the biceps attaches here along the scapula. Okay? So it's going to cross both the shoulder joint here, and then, look, it's attaching to the radial, so it's crossing the elbow joint. So that when the biceps contracts, it may cause movement at two different joints. It can cause flexion at the shoulder as well as the elbow. Okay. Now, is this biceps attachment, is it direct or indirect?
Indirect.
It's indirect, it uses tendons.
So if it uses a tendon at either point,
A tendon or an aponeurosis, and usually if it's on one side, it'll be on the other side.
okay.
Then it's indirect. Now, we said that in terms of skeletal muscles, the fibers are long and cylindrical. Multiple nuclei located beneath the cell membrane. Look here, we threw in a new word here, sarcolemma. Yeah, sarcolemma. Sarcolemma is a cell membrane along a muscle cell. So we give it a more specific designation. We call it a sarcolemma instead of just a muscle cell membrane. Okay. Fibers can vary quite a bit in both diameter and length. Okay. Now, the cytoplasm also has a special name. Instead of calling it cytoplasm, we call it sarcoplasm. You know, you're starting to get the feel that this term sarco is a specific designation for muscle, aren't you? Yeah, I can tell. You're picking up on that. That's good. That's a good thing, as Martha Stewart would say. Okay, now, early on in A&P 1, we said something very, very special. We said that glycogen, the storage form of glucose, is found in liver and skeletal muscle. You remember us talking about that? We said that animals store glycogen in liver and skeletal muscle. Now, that glycogen is stored as granules called glycosomes. So that muscle cells possess glycosomes, and they also possess an oxygen-binding protein called myoglobin. It's very similar to hemoglobin found within red blood cells. It's similar from the standpoint that it binds oxygen, and when it does so, it becomes bright red, just like red blood cells. Okay? So it imparts a certain color or pigmentation to the muscle, myoglobin. Now, a lot of the organelles are your usual organelles, like mitochondria. You can imagine that contracting muscle requires a lot of ATP, so you have a high density of mitochondria, particularly in skeletal muscle. Now, you have an endoplasmic reticulum, just like you do in other cells, but in skeletal muscle cells, we're going to call it a sarcoplasmic reticulum. Okay, you see what I did there? I used that word sarco again. Okay. I don't know if y'all notice that. Sarcoplasmic reticulum. Okay. And then you're going to have some unique features, one of which is these rod-like structures called myofibrils. This is actually a model of a single skeletal muscle cell. Okay? Let's take a look. This is the sarcolemma. This is the cell membrane. So you say, well, what is this right here? What is this other covering right here? That's your endomysium. Okay, that's the sheath that covers a single cell. Now, if we look inside the cell, these rod-like structures are called myofibrils. Okay? They contain muscle proteins that form alternating dark and light bands. Okay? So it's going to be what gives the cell its striations. In fact, the alternating dark and light bands within one myofibril are in register with other myofibrils, so that the dark and light bands alternate not only along the myofibril, but across the entire cell. Yeah. Now, if we look here, what do you think this thing is right here? Just deep to the sarcolemma. That's the nucleus, yeah. What is, and of course, here's its nerve supply. Okay, this would be an axon terminal. You can see that this is a myelinated axon. Okay? It's forming a neuromuscular junction here. along the sarcolemma. So we're using some new terminology. We'll also talk about another unique feature, T-tubules. T-tubules are simply invaginations of the sarcolemma that dive deep into the cell. So this is a model you'll want to become very, very familiar with. This is a model you'll want to embrace, to give it a little love. Okay. You think it will be on the practical?
Definitely.
Oh, you are too, yeah, yeah.
What should not be on the practical? And
It's not.
Ooh,
No, I can guarantee you it'll be somewhere on the practical. So we come back to this concept of myofibrils. Myofibrils. These are densely packed rod-like contractile elements. Look how rod-like they are. Okay? The cell is packed full of these myofibrils. Okay? And we said it houses proteins. These are 4 big proteins within muscle. Myosin, actin, troponin, and my personal favorite, tropomyosin. Actin, myosin, troponin, tropomyosin. Okay?
Why do you look at me when you pronounce words?
What's that? Say it again?
So why do you look at me when you pronounce words?
Oh, I look at everyone. I look at, yeah, everyone. Okay? So you can tell that the myofibrils make up most of the muscle volume. And we said that the fibers, those myofibrils made up of those proteins are arranged such that you have an alternating pattern of dark and light bands. Now, we're going to call the dark bands, dark is spelled D-A-R-K. We're going to call those A bands. And we'd call the light bands, well, let's call those I bands. And as an easy way to remember it, just look at the second letter.
Look at that.
Yeah, yeah. So these myofibrils are made up of an alternating pattern of dark and light bands. So here we see a diagram that looks a lot like that structure right there. You see that inside of the cell, if we pull back the sarcolemma, there's a nucleus, but what we see mostly is these myofibrils. Now buried between them, you see lots of mitochondria, because we need energy. And then along the myofibril, you see this pattern of dark and light bands. Now to show you something that many of you may not have noticed if I wouldn't point this out. Here's your dark band. Right next to it, this lighter staining band. And then running down the center of this light band here, see this dark line?
Mhm.
That's called a Z-disc. A Z-disc. Now, why am I making such a big deal over these Z-discs? Because this portion of the myofibril from one Z-disc to the next, and again, from this Z-disc to the next, and again, this portion of the myofibril is called a sarcomere. A sarcomere. The sarcomere extends from one Z-disc that's running down the center of the light band to the next. Oh, and lo and behold, look here. We have a slide that's dedicated to the sarcomere. It's the smallest contractile unit of a muscle. It's the region of a myofibril between two successive Z-discs. Okay? And that sarcomere is going to be made up of myofilaments that are formed of contractile proteins. Okay? You have two different types of myofilaments that make up sarcomeres. You have thick filaments and thin filaments. And the thick filaments extend the entire length of an A band. Okay? So an A band, let's suppose that this is the dark band, the A band. Well, spanning its entire length from front to back is going to be thick filaments. Okay? Thin filaments extend across the light band, the I band, and then partly into the A band. Now this can get kind of confusing at the beginning. It helps if you have your colors. Colin, did you bring yours?
No.
Okay. You can borrow Jacob's. Because the truth is, we're going to have a little contest for extra points. And the person who in the end does this the most accurately and the most nicely is going to win some big points.
You know, make it red.
Okay. All right, so we said the sarcomere is made up of thick and thin filaments. I'm going to use this dark color to represent thick filaments. Watch here. There's a thick filament. Here's another thick filament. Once I get going, it's hard for me to stop. All right, so those are all thick filaments. I'm going to use red. No, Ned. Pink for my thin filaments. Now, earlier I said that the Z-disc extends down the center of the light band. Now, what it's doing is it's tethering, it's holding together all these thin myofilaments.